Well, hi everybody. Welcome to the review video for the Bacterial and Viral Genetics lectures. Lost track of what week we're in. I think this is week 9, but whatever week it is, that's the material we're going to be covering. This is going to be a long one. I got to warn you ahead of time. I'll give you some points at which you can pause, and it would make sense to pause, so you can break this up over multiple watching sessions if you want to. We're going to be talking a little bit about the bacterial genetics, defining all of the Fs, F minus, F plus, F prime, Hfr. We'll be talking about rolling circle replication and how we use that to map genes with high-frequency cells, or Hfr cells. And then we'll have a good pause point if you want to take a break. And we'll move on to the viral stuff, talking about the lytic and lysogenic cycle, generalized and specialized transduction, and we will wrap up with Benzer's experiment, which is probably hands down the most reported confusing concept from these two lectures, and understandably so. It's the highest level way of thinking about this. So to start off, we can talk about two bacterial cells that differ in the fact that they have or don't have a single plasmid. Now, to be fair, each of these bacterial cells has its own circular genome. That's not what we're talking about. It's just that this cell has an additional circle of DNA, a much smaller circle of DNA called a plasmid, and the other cell does not have a plasmid, so there's a plasmid missing from the cell on the right. Now these plasmids, the ones that we talk about here in this class, are pretty specific in that they are called Troplasmids, or Troplus Plasmids, and Tra stands for transferable. In other words, the plasmid and the plasmid itself can cause its own migration or transfer to another cell of the same generation, in the same time. So this is called horizontal gene transfer, or sometimes lateral gene transfer, where those two words are meant to capture the same thing. DNA is moving across a single generation, not generationally from older to younger. All of us will have engaged in, at some point or another, all of us are the beneficiaries of vertical gene transfer. Vertical gene transfer is when genes are transferred from one generation to the next. Even if you don't have kids, you are the beneficiaries of vertical gene transfer because you got your genes vertically from the generation above you, from your parents. And if you do have kids, you continue that vertical transfer of genes. So vertical gene transfer is pretty boring. It's sexual reproduction. It's how we get genes to the next generation. Horizontal gene transfer is the transferring of genes to a different individual of the same generation. If I were to give you my genes or you were to give me yours, we would engage in horizontal gene transfer. That's not possible in humans or larger metazoans, but it is possible in bacteria. They can transfer their genes laterally or horizontally. And the way that that works is that there are proteins on this plasmid, or I should say it more correctly, there are proteins encoded by genes found on this plasmid that allow the cell to make some structures to allow this plasmid to be transferred. So we say that this cell is fertile in more of kind of the sexual reproduction way of thinking about it, in that it can transfer its DNA to another organism. That DNA can be traded, and alleles can be traded between organisms. So if this is a fertile cell because it has a plasmid, we say that it's F+. And this would be an infertile cell. In other words, there's no plasmid for this cell to transfer. It can be the recipient of a plasmid, but it can't donate a plasmid to another cell because it doesn't have a plasmid in the first place. We call that an F- cell. So essentially F+ means you have a plasmid, and F- means you don't. One of the plasmids, one of the genes on the plasmid, is for a protein that controls the formation of a tube, a hollow tube, that then connects these cells. That tube is called a conjugation bridge. Now, it takes a while for these conjugation bridges to form. They're pretty elaborate structures. So before a conjugation bridge can even be established, the F+ cell must first put out almost like protein cables, protein binders, that allow it to hold on to the F- cell that it's going to trade DNA with. And these cables, these non-hollow protein cables, are called, individually, they're each a pilus, and together they're pili. So again, those proteins for making the pili are encoded on the plasmid as well, and this is what makes the plasmid transferable. The proteins on the plasmid allow the cell that holds the plasmid to make the proteins necessary for a sex pilus, for sex pili. It allows the cell holding the plasmid to make the proteins necessary for a conjugation bridge, this hollow tube, and now conjugation, or lateral gene transfer, can occur. So we're going to go back to rolling circle in just a minute, but suffice it to say at this point that the plasmid is copied and transferred into the recipient cell. Now the recipient cell has its own copy of a plasmid. It's not as though this plasmid moved into this cell and it's no longer here. The plasmid is copied, and the copy is moved into this cell, and now this cell is an F+ cell as well. The sex pili and the conjugation tube will eventually break down over time. They're pretty fragile structures. And now each of these cells goes off, and they can repeat the process with another F- cell. This cell remains F+, and this cell became F+ because it had a plasmid transferred to it. So those are the essences of gene transfer and the definition of F+ and F-. Let's zero in on rolling circle replication before we go on to Hfr cells and F' cells. So for rolling circle replication, I'm going to zoom in now on what I drew before. So this is going to be the plasmid, nice and big. Here's the cell membrane of the F+ cell, and here's the conjugation tube going to the F- cell. So the F- cell is somewhere off the screen here to the right, and this is the F+ cell blown up nice and big. And there's even a genome, a giant genome here, a circular genome that we're not even seeing. This is the plasmid. So the reason why I draw the plasmid in that way, I put a circle and then I kind of put this ruffle around it, is to emphasize that this is double-stranded DNA. Like all DNA, plasmids are double-stranded, and those strands are related to each other through complementary base pairing, something I'm sure that you've encountered in other classes already, probably even before college. When DNA is double-stranded, wherever you have an adenine on one strand, you have a thymine on the other. Wherever you have a cytosine on one strand, you have a guanine on the other. These two bases are held together through hydrogen bonds. DNA that's double-stranded is complementary. If I give you just one strand, you can automatically tell me what the other strand should be, because C's always go with G's and A's always go with T's. So in rolling circle replication, what happens is one of these two strands is nicked, so it's no longer continuous. And through the action of proteins that, of course, are found on the plasmid itself, that nicked strand is unwound from the other one. So here I have it unwound a little bit. Now I want to emphasize that if we were to put in some sequences here for this nicked strand, the corresponding complementary sequences would be on the other strand of the plasmid that's left behind, right? Because this thymine was just base paired with this adenine, the cytosine with that guanine, the guanine with that cytosine. We've just kind of peeled one strand away from the other. And now what's going to happen is this circle of plasmid DNA, well, what's the name of the replication technique? Rolling circle. This circle of DNA of the plasmid is going to roll. It's going to roll, in this example, in the counterclockwise direction, and as it rolls, we're going to peel more and more of this single nicked strand off of the other intact strand of the plasmid. So let me kind of capture what's going to happen after that rolling. So we've rolled a bit. Now that nick, where we nicked it, is here, let's say. So here's the rest of the DNA that's still on its corresponding strand, but because we've rolled and peeled away that DNA, even more is going through the conjugation tube. Even more single-stranded, nicked, complementary DNA is going through the conjugation tube. I think that's the sequence that I put before. That means the corresponding sequence here would be A, G, C, G. But we've rolled, so all of these bases are correlated to and are complementary to the bases that are here. And let's keep rolling. Let's dial that circle of DNA even more counterclockwise. So here's that Nic point. And I can't even capture all of the DNA that's in the conjugation tube because some of it is already headed over to the F- cell. And if we had that A, G, C, G here, I can't even put in the corresponding complementary base pairs because they're over here. They're off the screen. And if we roll just a little bit more, and many of you can probably already see it coming, then we've rolled that one strand completely off of the other, and all of that unwound, nicked single strand of DNA is making its way through the conjugation tube to the F- cell. Now this is now single-stranded DNA. Single-stranded DNA is not very stable in a cell, and so the cell is going to immediately recognize that this is single-stranded. It's going to say, oh no, I got to do something about this. Let me replicate this single strand and make a double strand, because as the cell, I know that I should always have double-stranded DNA, and that green ruffled strand is the new corresponding complementary strand of DNA that's made against the existing circle of the previous plasmid. And the cell can easily do this because wherever there's an A on the new strand, there was a T on the old. Wherever there's a G called for on the new strand, it's because there's a C on the old, etc. So this cell remains F+. Yeah, one of the two strands of the plasmid was just made, but it's a double-stranded plasmid once again. It's intact. This is an F+ cell. Let's zoom over to what's been going on in the F- cell all the time. So here's the rest of that conjugation tube. Here's that cell. And in is coming that single strand of DNA that was peeled off of and rolled off of the F+ cell. So let's give it a little bit more time for that DNA to fully migrate into the cell. So now here is that single strand of the plasmid. And the cell freaks out because not only is single-stranded DNA bad in the cell, but single-stranded non-circular DNA is the worst. So the cell's going to freak out, and it's going to stitch these two loose ends together. So I'm a prokaryotic cell. I need circular DNA. Whew, let's make a circle. And then it's going to recognize that that circle of DNA is single-stranded, and it's going to fix that by just putting a T wherever it finds an A wherever it finds a T, a G wherever it finds a C. It's going to make that single circle of DNA double-stranded, and lo and behold, that's a second intact plasmid. This cell just became F+. So that's rolling circle replication. Rolling circle replication, again, to summarize very quickly, is nicking one of the two strands of an existing plasmid, taking that nicked strand and unpeeling it from the other strand by rolling the plasmid physically and feeding that single nicked strand of DNA into the conjugation tube or bridge so that it can make its way over to the other cell, be recircularized first, be copied second, and therefore become an intact plasmid to end up with. So we've defined F-, we've defined F+, we've gotten to rolling circle replication. There's some good stuff here. Hopefully it's clicking in a little bit better than before. Now let's move on to Hfr cells. I'm just going to put one big cell here. Here is the genome. That's the circular genome of the cell. And let's put our plasmid in. Let's put our plasmid in green here. There's the plasmid, much, much smaller. Sometimes what happens, now not commonly, rarely, but sometimes what happens is there will be a recombination event between the genome and the plasmid, a swapping of genetic information, but not so much a swapping as more so a Philadelphia kind of translocation, where the entire plasmid will be recombined or integrated or absorbed into the genome. So let's draw what we mean there. As a result of this recombination event, now remember, the plasmid's DNA is much fewer than the genome. We'll end up with that, where the plasmid sequences are still present in the cell, but the plasmid is no longer independent. Instead, that plasmid DNA, those plasmid sequences, have been fully integrated into the host cell's genome. Now, this is not an F+ cell anymore. An F+ cell is when the plasmid is independent. So when we have this kind of integration event, we refer to these cells as Hfr cells, or high-frequency cells. Now, what is it high frequency? What do we mean by high frequency? Well, what we mean is that these types of cells, and only these types of cells, can transfer genes, genomic genes, bacterial genes, not just the plasmid, but can transfer bacterial genes laterally. They can give their own genes, black DNA, DNA from the original chromosome, to another cell of the same generation. Well, how would that work? Well, the first thing that would happen is that this cell would make some sex pili. And it can do that because there are genes for making a sex pilus on the plasmid DNA, and the plasmid DNA is still on the cell. So let's put out some of those protein cables. And the next thing the cell would do, because it has genes for the proteins that do this, is create a conjugation tube or a conjugation bridge to connect to that neighboring cell. So let's do that. Still, all that information is on the plasmid, and the plasmid DNA is still present in this cell. And then the plasmid is going to try to transfer itself, because that's what troplasmids do. And it's going to first call for a nick. Oops. Wrong color. So there's the nick, just like before. No new material here. And then that circle is going to roll, just like before. So let's draw that. And I'm going to really roll it this time, because I want to make sure that we get to the concept we're after. And you already saw rolling circle replication. So we've really rolled a lot. We started with the integrated sequence here. I've rolled quite a bit counterclockwise. And so this DNA is still associated with the genome. But we've unwound a lot. And the very beginning of that unwinding was, of course, the plasmid DNA. So let's look at what's happening here. The plasmid DNA is doing what it has stupidly been programmed to do. It has initiated conjugation. It's trying to transfer itself. The plasmid DNA is in green. But because the plasmid DNA had previously integrated into the host genome, the entire chromosome is undergoing rolling circle replication. The entire chromosome is winding and unraveling. And we have a lot of genomic sequences making its way through the conjugation tube. That's true lateral gene transfer. Black or host genomic DNA in this picture is making its way through the conjugation tube and will be transferred to the other cell. Now it's important to note, mentioned it earlier, conjugations are fragile. They rarely last the amount of time that it would take for the entire genome to be transferred. That would take about 90 minutes. The slightest disturbance, the slightest bump or knock on the table of the sample undergoing conjugation would have this whole thing break down. So usually, in a time-dependent manner, sooner or later, this conjugation tube and the sex pili will just break down. And whatever you've transferred thus far will get transferred, but other sequences will not make it through the conjugation bridge. If we look at the end result of that, what we see is that the samples, or to be more precise, the genes, the genomic DNA that is transferred first in this Hfr transfer is DNA that was closest to the plasmid insertion site. Makes sense. The plasmid DNA gets transferred first because that's what the plasmid was programmed to do. The rest of the genomic DNA is actually just going along for the ride. So a very, very short conjugation will result only in the nearest, most neighboring genomic DNA being transferred. Whereas if we could get a conjugation to go all the way to the full 90 minutes, we would actually get the DNA from all the way on the other side of the genome. We would get the DNA that flanks the plasmid on the other end. Most of the time, we get something in between. We'll get the DNA that goes closest to the plasmid at the nick site first, and then if we have longer and longer conjugations, we'll get more and more genes further and further into the chromosome. And that's going to become important in just a moment when we talk about gene mapping with Hfr strains. So hopefully we're building our concepts here. Nothing new in an Hfr strain. It's just that the rolling circle replication includes and encompasses genomic DNA because the plasmid had integrated first. Before we talk about gene mapping with Hfr strains, let's take that quick detour, though, just to define our last term. Let's go back before our conjugation, but the integration has occurred. So this is an Hfr strain because the plasmid DNA in green has integrated into the genomic DNA in black. Now, just as we have the rare event of the integration, we can also have the rare event of what we call excising or excitation, excision, of the DNA. And that just means that essentially the recombination reverses, where the plasmid pops back out and goes back to becoming independent. Now, sometimes those excisions are clean, as I've drawn it here, and we just revert back to being an F+ cell. But sometimes those excisions are not clean. Sometimes the plasmid makes a mistake as it excises, and it actually cuts itself out a little bit off-center or a little bit imprecisely. And when that kind of cut happens, what we make, or what we excise, is a plasmid that has a little bit of genomic DNA as part of it now. Because when it cut itself out, it stole a little bit of the genomic DNA here in black by accident. So this is completely unintentional. This is an F' cell. An F' cell, by definition, had to have been an F+ cell that went HFR due to a recombination or integration event, and then had an improper excision to take with it a little bit of bacterial DNA. Now, F' cells are also capable of true lateral gene transfer because the plasmid DNA is still intact, which means we can make our sex pili. Plasmid DNA is still intact, so we can make a conjugation tube. Plasmid's going to do what the plasmid always does, call for a Nic, call for rolling circle replication. It's going to transfer itself completely through the conjugation bridge. It has the time to do that, leaving a single strand version of itself behind. But, look what's happening. Right here. What's going across to the F- cell? That little bit of genomic DNA that was stolen from the excision. That's our lateral gene transfer right there. When we're transferring genomic DNA from one cell to another, we're engaging in true lateral gene transfer or horizontal gene transfer, and it's because of that improper excision event. It's because of that F' nature of the cell. All right. I am having fun. I thought this was going to be long. We're already at 22 minutes. We've gotten through quite a bit. And I like this stuff. I always think I'm not going to enjoy this week because of bacterial and viral genetics. Every single year I'm like, here comes that week. And then I have a blast. I find this stuff to be far more interesting than I appreciate. Anyway, I digress. So let's go back to an HFR strain. Here's the the cell. Here's the genome. I'm going to be a little bit proactive here and leave a space for myself to put in the plasmid DNA. The integration has occurred. Again, we are HFR. And let's imagine that the Nic is going to be right here. And let's give these genes some letters, some regions, just like we did for the chromosomal rearrangement. So we're going to identify large regions of the chromosome by letter designation. So we'll call this region A, we'll call this region B, C, D, E, and F. And if we nick here and we're going to unroll in this direction, hopefully you can see that the very first region that would make it through the conjugation tube, and really not need all that much time to do it, will be region A. And if conjugation is allowed to go on even longer in some cells, other cells will give us A and B. And if conjugation goes on even longer in a subset of cells, we might pick up C, we might pick up D, we might pick up E, and if we have the luckiest of luck, and we have a full 90 minutes of uninterrupted conjugation, we could very well get the whole damn chromosome, all regions A through F. And we will have achieved gene mapping, because now we'll know that region B lies between A and C, region E lies between D and F, C is in the middle of B and D. We would map these genes, map these regions. But, unfortunately, that's not likely because conjugations are so fragile. So the other thing that we could do is take multiple different cells and hit them with plasmids that integrate in different regions. So let me kind of give myself a little bit more room here to tell you what I mean. So let's imagine a second cell, identical cell in terms of its genome, so the same exact genome, and the same exact genome, so the same exact regions. But now, let's say that the integration on this cell actually occurred here. So this is a different kind of HFR strain. The plasmid went into a different spot. That's why I gave it a different color. The plasmid went into the region here. That's the blue plasmid. And let's say that we are going to nick this plasmid here. So we can begin to make some predictions. Well, we already said in this strain we're going to get A, then B, then C, maybe we would get D. And that would probably be the longest we could hope for. We would never see E and F in this case. Here, we're going to nick here, we're going to unroll in this direction. And so the first region that we're going to pick up from this HFR strain is D, and then E, and then F, and then A, and we probably wouldn't get anything much longer than that. But if we take the cells away and we just look at the gene transfer, what we notice is that based on the regions that we got from these two different high-frequency strains, the one that had the green insertion site and the one that had the blue insertion site, we have enough overlap to actually map the entire chromosome. So if we put a circle down and we say, okay, well, this green plasmid, it inserted right here. And actually, let's just give that an arrowhead, if we would, to represent the direction of gene transfer. The point of the arrow is the nick point. So we're actually going to transfer away from the arrow. You can think of this as the head of the snake, the point is the nose of the snake, and the transferred material is the body of the snake lying behind the head. For for the green insertion site, we get A, B, C, D. We don't know what's going on the rest of the genome. We have it as a circle because we know the genome has to be circular, but we don't really know the rest of what's going on. Then we look for overlap. We say, well, wait a minute. We have a D in our second insertion site. But what we know is next to D already is C. So this must be going in the opposite direction. The unrolling must be going in this direction, and that's why we have D, E, F. Oh! We've closed the circle. There's the A we saw before. So if the blue plasmid is unrolling in this direction and we picked up D first, then that arrowhead must be right here. Remember, the point of the arrow is the nick, and so the first thing we would pick up is the body of the snake, D, E, F, A, and we've mapped the entire chromosome. This overlap gives us the entire chromosome mapped. And that's the essence of HFR gene mapping. So we'll have some practice problems with those along the way, but I'll just diagram for you very quickly how we can go in either direction there. So let's give ourselves a bunch of genes. I'll do one more. And they won't always be in alphabetical order. I'm just doing that to make it easier for us to see it. These would be scrambled on a quiz or an exam question, but the principles are the same. And let's put a few insertion sites here. So that's 1. That's 2. And that's 3. Let's give ourselves really good coverage. That's 4. And let's say that's 5. So 5 different plasmids in 5 different cells. So to be fair, we're kind of putting it here as a composite, but this would be 5 different cells with 5 different plasmid insertion sites. You can think of this as the green that I drew before, the blue that I drew before, and maybe red, orange, and purple. I'm just not using the colors, I'm using arrowheads and numbers instead. And now we can make some predictions. So for the high-frequency strain number 1, where the plasmid went in this point, if this is the nose of the snake, the very first gene we should see is C, and then D, and E, and F, and G, and maybe we last long enough for H. And for 2, we would get, if this is the point of the snake and we're going away from the snake, we're going towards the body, we would get F and E and D, and maybe we just stop there. For 3, if this is the point of the snake here, we get I, we get H, we get G, F, E, maybe we pick up D, and that conjugation breaks down. For number 4, here's the nose of the snake, we're going away from it, we get K, J, I, H. And for 5, we're actually going in the opposite direction, we get K, L, M, A, B. Alright, so let's see, and we'll get one more. Let's say we get C. So that's going in one direction. That's you're being given the chromosome, the chromosome of the bacterial cell, and you're being given the Hfr insertion sites, the plasmid insertion sites, and you're being asked to predict the mapping order of the genes, or the transfer order of the genes, and you can do that. Just start at the nose of the snake and go backwards. But what you would really be asked to do is the opposite. You would be given the gene transfer pattern for a number of different Hfr strains, and you would be asked to map the entire chromosome based on those transfer patterns. And the procedure for doing that is always the same. Start with a circle. That represents the chromosome. Start with an arrowhead. That represents the first plasmid insertion site. And draw in your gene transfer, making sure that you're putting in the body of the snake. Don't put those genes ahead of the nose. Put them by the body. So C transferred first. That means it's closest to the plasmid. Then D went, then E went, then F went, then G went, then H went. And I'm going to look at my map and look for overlap. F, E, D, well, that's here. F was transferred first. I'm going to make F, E, D the body of my snake. Here's the nose of my snake. This has to be plasmid insertion site number 2. I go on to number 3 and I look for overlap. Well, I have overlap here at F, E, D, G, H. It's the I I'm missing. The I comes next to H. The I gets transferred first. And so I'm going to put my arrowhead in for number 3 and double check it. If this is the nose of the snake, it should be I, H, G, F, E, D, I, A. Yep, perfect. Then I'm moving on further still, looking for overlap. So I have H, I have I. This says that J comes after I and K comes after that. But K was transferred first, so K needs to be closest to the head, forming the body of my snake for number 4. And finally for number 5, I've got my K. Next to K on my map, I've got a J. So this L, that must be on the other side of K. L, M, A, B, oh, and there's the C. I closed my circle. I've come all the way around in my map. I just have to put in the insertion site for number 5. K is the first thing to transfer. There's the nose of my snake. There's 5. That should be K first, then L, then M. And so there's my genomic map. You'll notice it's a little bit skewed from what I had before. I had the A up here, but that's okay. It doesn't matter how it is rotationally. It just matters that all of the genes are mapped correctly and their orientation to each other. In other words, they're all next to each other in the right way, and we have those plasmid insertion sites correctly mapped as well. So just a quick example of what you would be asked to do with this moving forward. All right, so as promised, this is your good pausing point. If you've had it and 34 minutes is enough time, feel free to stop the video now and come back to it later. We're going to move on to the viral stuff next. So on to viral aspects here. Again, my bacterial cell. And there's my bacterial genome. No plasmids in this discussion because this is the viral genetics piece. But what we do have is an infection. So here's one of those moonlander viruses, one of those beautiful viral heads, and it has its own viral DNA that it is going to inject for infection. So one of the mechanisms of infection can be that the viral DNA is injected into the host cell, and it immediately gets to work. It hijacks the host DNA replication machinery to make lots and lots of copies of viral genomes. Some of the genes on those viral genomes encode the proteins of the heads of those shells, those moonlander shells. And my apologies for my horrible moonlanders. You all know I'm a very bad artist. So now we've made viral particles. All of this is happening in the host cell, using the host's machinery to do that. This will continue on for so long that this cell will swell and swell and swell, and it will burst under the pressure of all of these viral particles that are filling this cell. And when that cell completely bursts or lyses, those viral particles are released, and they scatter into the environment to infect the next cell and repeat the process. Very, very quick and easy life cycle. And because it hinges on lysing the cells, this is the lytic cycle. And that's it. That's the lytic cycle. For the other life cycle of the virus, there's the cell, there's the genome, there's the moonlander, we have the infection, the viral genome is inserted, so again, very much like the previous life cycle, but in this case, the viral DNA, just like a plasmid in our Hfr strains, is going to recombine or integrate into the host genome. And it's going to lay there, dormant. And whenever this cell replicates its genome in an effort to undergo cellular reproduction, and it makes a brand new copy of itself, what's it also copying? The viral genome. Every time this infected lysogenic cell copies itself, it replicates the viral genome. So the virus still spreads. It just does so stealthily. It does so quietly. This is the lysogenic cycle. The lysogenic cycle is when the viral genome has integrated into the host genome and it lies there dormant, and it is spread passively by the host cell when the host cell replicates its own genome. Now, to be fair to the lysogenic cycle, sooner or later, in the life of any cell, that cell begins to experience negative stimuli. Maybe it begins to starve. Maybe it has picked up oxidative damage or UV damage. Maybe it's just an old cell and begins to break down. But whenever this cell begins picking up negative signals that its life is somehow in jeopardy, the virus, being the selfish bastard that it is, jumps ship. It reverses its lysogenic stage, and like that plasmid, it excises itself from the bacterial genome, and then it goes lytic, quote unquote. It hijacks the host machinery to make lots and lots of copies of itself. It uses the genes on its viral genome to carry out the assembly of the moonlander viral proteins. The cell will eventually swell under the pressure of this, and that swelling will cause this cell to lyse and those viral particles to be released. So with rare exception, all lysogenic cycles end with a lytic phase, as the virus removes itself and escapes from the dying cell before the cell has a chance to die naturally. But lysogeny, the lysogenic cycle, gets at this dormant phase where the virus is just lying there passively. And our own viruses have this. So the common cold, coronavirus, these are lytic viruses. They cause damage immediately upon infection, and it's the damage that causes the disease. That damage is basically the necrosis of cells, the bursting of cells, and that's what gives the symptoms of the disease. But we also have lysogenic viruses. They're not technically lysogenic, they're retroviruses, but the process works roughly the same way, in the sense that the virus has a way to go within our genome, to go into our actual genome, to integrate the viral DNA into our genome and hide there dormantly for a long time. Herpes simplex virus, HSV, is an example of this, as is HPV, and even HIV can all undergo this. So you can be infected with these viruses, but not have any symptoms because they're lying dormant, integrated into our own genome, and then they emerge later on when we experience cellular stress. So that's lytic versus lysogenic. Now let's get on to how we see horizontal gene transfer of bacterial DNA with these life cycles. So let's talk about generalized transduction first. Here is our bacterial cell. There is the bacterial genome. We've undergone infection, so we're in the middle of a lytic infection. Generalized transduction is typically associated with the lytic cycle, which is why we review the lytic cycle first. But as this cell becomes sick, as this cell begins to die under the stress and the overwhelming taxing of its physiology based on the infection, part of what happens in the necrosis of any cell is that the genome spontaneously fragments. Double-stranded breaks are no longer being repaired because the cell is dying, and so the genome breaks into fragments. What happens in generalized transduction is if a particular fragment of genomic DNA is close enough to a moonlander, the moonlander will scoop up, will vacuum up that bacterial DNA instead of vacuuming up viral DNA, which is what it should do. Because again, these viral particles are just as stupid as anything else. They're just following a program. They're empty viral shells with DNA motors on them, and they're just looking for small fragments of DNA that they can scoop up and load into their viral head. Now, as this process continues and the cell eventually bursts under the strain of all these viral particles, yes, we will be releasing normal viral particles into the environment that can continue the infection, but we will also be sending out into the environment these very, very unique pseudoviruses that are a fully functional viral shell, but what they contain is bacterial DNA. Bacterial DNA that upon a quote-unquote infection of the next cell, when that DNA is injected, you've just encountered horizontal gene transfer. This is bacterial DNA from one cell being injected into a another bacterial cell. It's the transfer of genetic material from one individual to another in the same generation. That is horizontal gene transfer, lateral gene transfer, but it's being transduced by a virus. So this is generalized transduction. Why is it generalized transduction? It's generalized transduction because the fact that this fragment came from this region was random. The genome had fragmented, and it would be just as likely and just as possible that this fragment be transferred in this way, or this fragment be transferred in this way, or this fragment, or this fragment. It was random chance that this fragment happened to be close enough to this viral shell that it's the one that got hoovered up and scooped up into the viral shell. Generalized transduction can transduce or transfer any region of the bacterial genome. It is random, it is based on the lytic cycle, and that general word means any old DNA from the bacterial DNA can be transferred. There's nothing special about it. Well, I choose my words carefully, don't I? I said there's nothing special about it. So what's so special about specialized transduction? Well, if it's transduction, that means that the DNA is being transduced by a virus. So we know a virus is involved. So there's our virus. And if it's transduction, then it's lateral gene transfer. So we're also going to have a bacteria involved. And we're going to have infection, and we're going to get viral DNA into the cell. But for specialized transduction, that DNA actually integrates first. So specialized transduction requires the lysogenic cycle. Key point. That DNA will lie dormant there for a long time, and it will be replicated passively, but sooner or later it's going to get a signal that the cell is struggling, and it's going to try to jump ship. But very much like our F' cells, see the overlap here? A lot of interplay of concepts in these two lectures. That's why we group them together in this week. Much like an F' cell, in some cases, when this virus excises in its effort to jump ship and go lytic, it makes a mistake. And it takes a little bit of bacterial DNA with it. And then these viral genomes are replicated, so they all have their little bit of bacterial DNA. And Oh, that's getting tedious to do the 2 colors. I'll do one more and then I give up. So that's all of our viral genomes, and then we're going to make the viral proteins that are going to hoover these up. And eventually the cell is going to swell under the pressure and burst, and all of these viral particles will go off and repeat the infection cycle. So if we zoom in on one of these viral particles, it is carrying a large piece of viral DNA and a little piece of bacterial DNA. That's our opportunity for lateral gene transfer. That's why it's transduction. That black bacterial DNA is being transferred from one individual of a generation to another individual of a generation, transduced by a virus. But we really didn't answer why it's specialized transduction, did we? So if we imagine that this was the original integration site, and the DNA that we are transferring here, transducing, is here, what we realize is that because this relied on an improper excision, yeah, it's possible that instead of making this mistake, which is what we made, it's possible that the mistake the virus makes is this one, and so it's possible we could transfer and transduce this DNA as well, but could we ever transduce this DNA, or this DNA, or this DNA in this mechanism? No. In order for specialized transduction to work, we need an excision mistake by the virus, which means it can only steal pieces of DNA that flank the viral insertion site. We can't transduce regions of DNA that lie far away from the viral insertion site because the virus would never include sequences so far away in this mistaken excision. So what's specialized about specialized transduction is that you're limited in the kind of DNA you can transfer. You can only transfer flanking DNA, DNA that lies on either side of the viral insertion site. So generalized transduction requires only the lytic cycle, and it can transfer any region of bacterial DNA. Specialized transduction relies on the lysogenic cycle and an improper excision event, and you can only transfer DNA that flanks the viral insertion site. All right, one more thing to cover, and then we are home free, the Benzer experiment. So the Benzer experiment is valuable to us for a number of different reasons. One, it brings us back to complementation, which is a concept that's critical for genetics, so it allows us to revisit that. Second, it's definitely directly relevant to what we're talking about right now, recombination events, gene linkage, and how we see different recombination events. It was done with viruses, so it's directly relevant to this week. And it has molecular connotations. In other words, it was really Benzer's work that showed us what the makings of DNA really were. Prior to this fundamental Benzer experiment, most scientists believed that the individual fundamental building block unit of DNA was genes. In other words, one gene was the smallest possible piece of DNA you could get. You couldn't break DNA into anything smaller than genes. And chromosomes were merely building blocks or Lego sets of genes linked together. And Benzer's work called all of that into question. Benzer's work basically proved that genes themselves are made up of even smaller building blocks, that DNA is made up of individual monomers that are separable and fundamental, and those monomers are linked together to make genes, and then genes are linked together to make chromosomes. Now, of course, now we know that those individual units are nucleotides, guanine, cytosine, thymine, and adenine, but that wasn't known at Benzer's time. So Benzer's work are very, very fundamental. So let's dive in and talk about how all these things come together into a wonderful story of recombination and the fundamental units of DNA on the molecular level. First, some ground rules. So Benzer used 2 different types of bacterial cells, B cells and K cells. Both of these are bacterial cells. Both of these are E. coli. They're just different strains, or you can think of them as different breeds of E. coli. So to make the analogy, you can think of these as dogs, but these are Pomeranians, my daughter's favorite dog, and these are Dobermans, or German Shepherds. So they're all dogs, but different breeds or different strains of dogs, and these are all E. coli, but different breeds or strains of E. coli. And they behave slightly differently, just like different breeds of dogs are quite different. The other thing we have is a bacterial virus that can infect E. coli, and this virus is named lambda, lambda-phage. That's the Greek letter lambda. If you were to spell it out, it would be lambda. And phage is an old-fashioned term for bacterial viruses. Bacteriophage is the old-school name for viruses that can infect bacteria. So lambda-phage is just a bacterial virus. This is a lytic virus, so it infects cells and it bursts them open. Now, B cells are promiscuously infected, K cells not so much. What we mean by that is essentially any lambda-phage can infect and lyse a B cell. K cells need intact lambda-phages. And to give this a little bit more meat, there's a region on the lambda-phage genome called R2. There are two genes in that region that we'll just call A and B. And in order for a K cell to be infected and killed by lambda-phage, both the A and the B gene need to be unmutated. But B cells die no matter what. So we can say that these always die, and K cells need wild-type R2, or unmutated R2, to die. And those are pretty much the ground rules. So just to recap, B cells and K cells are both E. coli, just different strains of E. coli. Lambda is the bacterial virus that's infecting these cells. There's a region on the lambda genome called R2, and it contains two genes, A and B. And if A or B or A and B are mutated, B cells will die and K cells won't. If the A and B genes are unmutated, B cells will die and K cells will die as well. Thems are the ground rules. The first thing Benzer did was he infected B cells and K cells with wild-type lambda-phage. So there was an intact A and B gene, and B cells died, and K cells died. No big surprise. And then he infected B cells and K cells with cells that had a mutation in the A gene but not the B gene, and B cells died and K cells didn't. Because in order for K cells to die, you need wild-type unmutated A and B. And Benzer wasn't surprised by this either. And then Benzer infected B cells and K cells with lambda-phage that had a wild-type unmutated A gene and a mutant B gene. And B cells died and K cells didn't. No big surprise, B cells always die, and K cells to die need a working A and B gene. No big aha moments. Then Benzer did a co-infection. And a co-infection is when two viruses land on a single cell simultaneously, and they both insert their viral genomes together. So a co-infection results in two viral genomes being in a single cell simultaneously. And here Benzer began to get clever. And he took one virus with a mutation in the A gene and no mutation in the B gene. And he took another phage virus with a mutation in the B gene and no mutation in the A gene, and he co-infected with those. So now, in one, you have a broken A gene but a working B gene, and in the other, you have a broken B gene but a working A gene. And he did that in both cell types. So broken A but working B, working A and broken B. And not surprisingly, B cells died, because B cells always die. But here, K cells died as well. And everybody in Benzer's lab got excited for a moment, and then it was just like a wah-wah deflation, because they realized that all they were seeing was complementation. And they already knew that complementation was possible. If we think molecularly, genes encode proteins. And in order to kill a K cell, you need a working A protein and a working B protein. That's what kills a K cell. And with the co-infection, the second viral genome had the information for making a working A protein, so A proteins were present in this cell. And from the first genome, we could make a working B protein. And since A and B proteins were present in a K cell, K cells died. That's the premise of complementation. It doesn't matter where those alleles are. It just matters that the information is in the cell and the proteins can be made. So K cells died due to complementation. Again, no big deal. So then, Benzer just kind of set out to prove that that's what was happening. And he took cells with an identical mutation in the A gene, took lambdaphage with an identical mutation in the A gene and no mutation in the B gene, and he did co-infections with that. And by identical mutation, I mean that what was wrong with the A gene was wrong in both of these phages. But there was co-infection. So here, you have identical mutations in the A gene, identical mutations in the A gene, and in this case, B cells died. Well, to be expected, B cells always die, and K cells didn't. And that's also not surprising, because there was no working A protein in these cells. Both of these alleles were mutated in the same exact way. The cell couldn't make A protein. K cells didn't die. To be expected. And then finally, Benzer took a chance. And it may be one of the most influential chances in the molecular revolution. Now, I'm speaking a little bit melodramatically there, because of course Watson and Crick's work was fundamental to the molecular revolution. It's also fair to say that Rosalind Franklin's X-ray crystallography work wouldn't have made what Watson and Crick did possible. Moving on to say the sequencing of the human genome has just opened so many doors, but it's Benzer's experiment, the experiment we're about to see right now, that laid the groundwork for that and got people thinking in that direction. Prior to this, everyone thought that chromosomes were linker locks or Lego blocks of genes, and this proved that couldn't have been the case. Outstanding, outstanding work by Benzer. And here's what it relied on. Benzer, taking that chance and throwing a dart at the board, did the last experiment where he took two different mutants in the A gene of R2, but those mutations were different. They occurred at different parts of the allele. One mutation was near the beginning of the allele, and the other mutation was near the end. And when Benzer infected B cells, co-infected B cells with these two mutants, they died. Not to be surprised by. B cells always die. And of course, complementation is not possible in this case, because this allele doesn't encode a functional A protein, and this allele doesn't encode a functional A protein, and so these co-infected cells should not engage in complementation, because neither of them encode a working A protein, and these K cells should not die, but they did. And no one could figure out why, until they realized that what must have been happening was something called an intragenic recombination. Think of interstates. Interstates are highways that connect states. You can get on the interstate and take 90 from Massachusetts to New York. You can get on 87 and take that from New York into wherever it goes. Interstates connect states. Intragenic recombinations are the recombination events we've been talking about all along. They're recombination events between multiple genes. Intragenic recombinations are recombination events that occur within a single gene. And though very, very rare, they happen. And like any recombination event, we're going to swap places. So we're going to take everything from the point of the recombination on, and we're going to swap it. So the intragenic recombination takes the mutation at the beginning of this A allele and swaps it up here to this allele, and takes the unmutated region here and swaps it down. The intragenic recombination is possible because of the co-infection, because both of these viruses are together in the same cell, and though rare, when it occurs, you recreate a functional A allele that can make the A protein. And you always had a working B allele, and these cells die. And that proved to Benzer and his lab that DNA must be made of something smaller than genes, because the break that's occurring here is occurring within a single gene. And if you can break a gene in half and recombine at that break point, then genes must be made of something smaller. And of course they are. They're made of nucleotides. Genes are strings of nucleotides strung together, and chromosomes are strings of genes strung together. But it's Benzer's work right here. It is these K cells dying upon this co-infection that proved that genes could not be the smallest and most fundamental unit of DNA. Amazing. So, that's Benzer's experiment, broken down as simply as I can make it. Hopefully you have a much clearer understanding of this material, all of the different Fs, F-, F+, F', and HFR. Hopefully you have a clear understanding of how rolling circle replication works and why it allows HFR strains to transfer their own genomic genes to a F- cell that they're conjugating with. Hopefully lytic and lysogenic cycles are more clear and how they relate to generalized and specialized transduction. And my goodness, hopefully you have some appreciation for the gloriousness of the Benzer experiment and how it set the stage for so much that came after it. Right here at the 1 hour mark, thanks for your attention. I hope you got a lot out of the video, and I look forward to your comments on what might still be unclear right now.